PCR or polymerase chain reaction refers to the reaction where the enzyme DNA polymerase in a thermocycler amplifies DNA from 1 copy to 2, 4, 8, 16, and so on. Before PCR, there was a time when to amplify DNA, you had to grow tons and tons of tiny cells. These lyrics of the PCR song refer to amplifying DNA in host cells. Amplifying in host cells involves cutting the DNA, ligating or joining it to a vector that carries it into a host cell where the DNA replicates. Then along came a guy named Dr. Carey Mullis, said he could amplify in vitro just as well. In some ways, amplifying in vitro, which refers to copying DNA outside living cells in the PCR process, is better as the thermocycler only needs a few hours rather than the days required for in vivo DNA amplification. But DNA amplification, whether in a cell or in a thermocycler, does not copy the whole chromosome, as a chromosome is too long to be replicated in a single reaction. PCR involves copying specific segments of about 1,000 base pairs. The key components in PCR are the DNA template with the sequence to be amplified, thermostable DNA polymerase, deoxynucleotide triphosphates, DNTPs, primers, cofactors, and a buffer. Primers are short DNA sequences that specify the chromosome region to be amplified by binding to the template complementarily. Thus, the sequence of the area where the primers will attach must be known. Luckily, scientists obtained and shared DNA sequences of humans and other organisms on databases we can all access. As seen here, this database shows the sequence of a single strand, but of course, since DNA is double-stranded and the second strand is complementary to the first, by looking at one strand, we can obtain the sequence of the second strand and design primers for both strands. Only with a primer for each complementary strand can we amplify the double-stranded DNA. Primers provide the free 3' hydroxyl the enzyme DNA polymerase requires to attach DNTPs. Since DNA polymerase can only add nucleotides to the 3' end, elongation only occurs in the 5' to 3' direction. To copy the double-stranded DNA, the 2 primers used in each PCR reaction have sequences that will make them bind to opposite strands of the template DNA, flanking the region to be copied with the 3' end facing the target region. The reverse primer is written as 5' ATAGCCG3' in the example shown, since DNA sequences are always written in the 5' to 3' direction. However, as shown on the diagram, the primer binds in the reverse direction on DNA. This example is simplified as actual PCR primers and PCR products or amplicons are longer. Primers are often 20 to 40 nucleotides, and authentic PCR products usually range from hundreds to about 1,000 nucleotides. These primers must be unique in the template DNA. There should be only one target site in the template DNA where the primer binds to ensure you amplify only the predetermined region you need for your purpose and not more than one region of the DNA, which would confuse the inferences from the PCR. Short primers have a higher chance of binding at multiple locations, so primers cannot be too short. To determine the possibility of annealing to other sites, we can compare the primer sequence to the sequence of the whole genomic DNA using a computer tool like BLAST or basic local alignment search tool. Forward and reverse primers should be designed so that they don't anneal to themselves or to each other at the temperatures used in the PCR reaction. If the primers do, they will be unavailable to bind to the template DNA, and the target region to be copied will not amplify. Long primers have a high chance of forming hairpins or dimers. Computer software can check if the formation of hairpins or primer dimers will occur. For the primer and nucleotides to bind to the template DNA, the DNA must be single-stranded, but then the primer and nucleotides have to bind and form double-stranded DNA. Thus, the thermocycler is programmed to raise, lower, and raise the temperature in cycles. Heating at about 96 degrees Celsius denatures or separates the double-stranded DNA. Cooling allows the primers to bind or anneal to their complementary sequences on the single-stranded template DNA. and slightly raising the temperature provides the optimum temperature for the heat-stable DNA polymerase to add nucleotides and synthesize new strands of DNA. Thus, one strand becomes two, two becomes four, and so on. The annealing temperature must be optimized so that primers bind specifically to the target region. If the temperature is too low, the primers may bind even though not all the bases in the primer are complementary to the template DNA in what is known as non-specific binding. The annealing temperature depends on the length of the primers and the GC content, thus the melting temperature. The annealing temperature is usually about 5 degrees below the melting temperature of the primer. The melting temperature is the temperature at which 50% of the double-stranded DNA becomes single-stranded and may be approximated using the following equation. Melting temperature is equal to 4 times the sum of G's and C's plus 2 times the sum of A's and T's in degrees Celsius. Only after many heating, cooling, and heating cycles will the target sequence be amplified. As seen during the first cycle of a PCR reaction, we are not generating the small amplicon. Copying template DNA forms long strands. These long strands will not disappear but will be much less at later cycles than the sequence targeted for amplification. The more template DNA you add to your PCR reagent mixture, the more long strands you will get at the end of the reaction, which is why in PCR you generally use very little template DNA into your PCR reagent mixture. There are many uses of PCR, such as PCR when you need to find out who the daddy is, PCR when you need to solve a crime. Interpreting PCR results will be discussed in a future video, but just briefly, one method analyzes them as follows. In a paternity case, the baby's DNA must be inherited from the father or the mother. From the DNA electrophoresis of PCR amplicons, we see that dad too is the biological father. While in a criminal case, DNA from a suspect and DNA at a crime scene, if identical, would strengthen the argument that the suspect carried out the crime. So from the DNA electrophoresis of the PCR amplicons, we see that suspect 3 was at the crime scene. In summary, the main points are: PCR amplifies a section of the chromosome between the primers, not the whole chromosome. Primers, short sequences of nucleotides that initiate DNA synthesis, must be unique in the genomic DNA, positioned correctly to copy the target region, and not form secondary structures at the temperatures used in PCR. A suitable annealing temperature must be used to prevent the non-specific annealing of primers. The amount of template used is small to prevent the presence of too many long strands compared to the required amplicon at the end of PCR to enable ease of result interpretation. PCR has many uses, such as identification and disease diagnosis. So let's check our understanding of PCR with a few questions. You may pause the video while you think about your answer. Question 1. PCR or in vitro DNA copying is based on the in vivo mechanism of copying DNA. Which statement on PCR and DNA replication is incorrect? A. DNA replication uses helicase to separate DNA strands and PCR uses heat to separate DNA strands. B. DNA replication depends on primers created by the enzyme primase. PCR also requires primers, but humans design these. C. DNA replication uses DNA polymerase to add nucleotides to extend the DNA strand. PCR uses a thermostable DNA polymerase to add nucleotides to the DNA strand. D. DNA replication has continuous and discontinuous strands, as does PCR. Statement D is incorrect as there are no discontinuous strands in PCR, as heating denatures the whole length of DNA, which can then be copied continuously. In cellular DNA replication, the enzyme DNA helicase opens up the DNA a little at a time. Since DNA polymerase can only add nucleotides to the 3' end, one section of each complementary strand can only be copied in short segments as the 2 strands of DNA progressively separate. Question 2. What would be the effect on the PCR reaction if there were no DNTPs in the reaction? A. PCR would proceed normally. B. Non-specific PCR of random templates will occur. C. The reaction will cease after a few cycles. D. The PCR reaction will not occur. The answer is D. The PCR reaction will not occur as nucleotides are the building blocks of DNA. Question 3. Imagine that 4 nucleotide primers would work for PCR. Select the primers which would amplify the region indicated in red. A. Primer 1 and Primer 3, B. Primer 1 and Primer 2, C. Primer 1 and Primer 4, D. Primer 3 and Primer 5. The answer is B. Primer 1 and Primer 2. A. Primer 1 and Primer 3 will not work because the primers bind to the same strand and extend the same strand. C. Primer 1 and Primer 4 will not work because Primer 4 is non-specific and also found in the region to be amplified. D. Primer 3 and Primer 5 will not work because the 3' end of the primers face opposite directions and will not copy the required area. Question 4. During PCR, the amplicon includes A. Only the sequence of the template between the primers, B. Only the entire sequence of the 2 primers combined, C. The sequence of the 2 primers and the template sequence between the primers, D. The sequence of the whole chromosome. The answer is C. The sequence of the 2 primers and the template sequence between the primers. Question 5. How many cycles of PCR does it take to produce a double-stranded DNA fragment of the desired length? A1, B2, C3, D4. As seen in the animation of PCR, the answer is C, in the 3rd cycle. Question 6. You use PCR to determine if you have the allele that increases your chance of getting breast cancer. You then choose also to check if you are at risk for developing Alzheimer's, another genetic disorder. What PCR ingredient would be different in these 2 tests? A. Template DNA, B. Nucleotides, C. Heat-stable DNA polymerase, D. Primers. The answer is D. Primers. Different genes cause the 2 disorders and will be located at different loci on the genome, and the amplification of different loci need different primers. Question 7. Complementarity of primer pairs at the 3' ends will result in what artifacts? A. Primer dimers, B. Large misprime products, C. Self-dimers, D. 5' end degradation of the primers. The answer is primer dimers as seen in the diagram. I hope this video has helped you understand the basics of PCR. Future videos will discuss interpreting PCR results and variations of the basic PCR process, such as real-time PCR. So do subscribe to be updated when new videos are uploaded. You may also want to explore previous videos to learn more about biology. Thank you.